Hello everyone, my name is Deepesh Joshi. Welcome to Chandu Biology classes. Today is the lecture 4 for cyclic and non-cyclic photophosphorylation, and in the previous lectures we have studied about the structure of PS2, PS1, and also we have learnt about the different photosynthetic pigments which are there. Okay, so in today's lecture we will be dealing with the cyclic and non-cyclic photophosphorylation, which is a very important topic. Most of the questions in the examination, whether it is your CUET PG, GAT-B, arises from this topic only. Okay, so yeah, so I am Deepesh Joshi, an integrated PhD scholar at ISC Bangalore, and I have qualified all these examinations. And in these lecture series, I will be focusing mainly on the GAT-B and CUET PG curriculum. Okay, so without any further delay, let's begin with the lecture. Okay. So, so today is the day when I will be answering that question which I asked in the first lecture. Okay. I hope you have watched all the lectures in this series. If you haven't, then please go back to the first lecture and watch sequentially. Okay. Now, so you know about the equation of photosynthesis, that is 6CO2 plus 12H2O in the presence of light give and chlorophyll give rise to C6H12O6, that is glucose, 6H2O and 6O2. So this is the equation with which each one of us is familiar with. But some people also write this equation 6CO2 plus 6H2O in the presence of light chlorophyll, it give rise to C6H12O6 plus 6O2. So you will say, sir, if I move the 6H2 here, it will be the net equation that you have written on the top, and that is more correct. Numerically, both of these equations are correct. But biologically or scientifically, this equation is more correct. Why? So till now we have looked into the splitting of water molecules. We have understood how water molecules split in the oxygen involving complex. So basically two water molecules, they give rise to what do you call as your four protons, four electrons, as well as one oxygen molecule. So two water molecules give rise to one molecule of oxygen. If if you have to yield six oxygen molecules, here you have to multiply this with 6. So you require 12 water molecules. So in this reaction, you are representing that evolution of oxygen is because of the splitting of water molecules. Okay, because of splitting of 12 water molecules, you are getting 6 molecules of oxygen. That is what is being represented by this equation. Okay, and by this equation, it is giving a wrong notion that 6 water molecules, when they are being like used up or they are splitted, they give rise to 6 oxygen molecules, which is a wrong representation that is being carried over here. Because people have done radio labeling experiments in which they radio labeled this oxygen of the carbon dioxide, and they identified that the oxygen that is evolved, that doesn't have any sort of radioactivity. But when the radio label this, when they use O18 instead of O16, and they radio label the oxygen of the water, they found that the oxygen that is evolved is again radio labeled. So that means oxygen comes from the splitting of water. To represent that correctly, we need 12 molecules of water in the LHS, so that you can represent that 6 O2 are released. So that is why this equation is more correct. I hope you got the answer. Like, we have the suspense from these lectures. Okay, I hope this is clear. Then, as I told you that we have two different sorts of reactions. In case of photosynthesis, one is a light reaction, another is a dark reaction. Light reaction is called as a photochemical phase, and dark reaction is also known as the biochemical phase. Okay, it happens, the site of light reaction, it is your thylakoid membrane, and the site of dark reaction, it is your stroma. In light reaction, ATP, NADPH are formed, In dark reaction, these are used up to make the food, that is glucose. In light reaction, photolysis occurs of water. Okay, in presence of light, water molecules they split to give rise to proton, electron, and oxygen. But in dark reaction, the carbon dioxide from the atmosphere is fixed. That is the major difference. Now, we will be dealing with something that we call as the process of photophosphorylation. That is the process by which ADP combines with Pi, that is inorganic phosphate, to form ATP, and using the energy of sunlight. That is what is called as photophosphorylation, guys. Whenever in our body, in mitochondria, there is something that is called as oxidative phosphorylation. Oxidative phosphorylation, because there we are oxidizing some compounds, okay, like NADH to give NAD plus, and we are using this energy of the electrons and the protons that are being formed in this oxidation reaction. We are using them to make ATP. So that is why that is called as oxidative phosphorylation. But here in case of plants, it is called as photophosphorylation, because it is happening in the presence of sunlight, and depending upon the path of electrons in the electron transport system, we have two types of what do you call as the photophosphorylation. So first one can be cyclic photophosphorylation, another is non-cyclic photophosphorylation. So let's look into them in detail. Before that, let's understand the exact, you can say, orientation of electrons and at the molecular level what is happening when light is falling. So as you all know, as I have told you, that you need to remember this Vibgyor, right? And as you move from top to bottom, your wavelength increases, your energy decreases, your frequency increases. Now, you can see that blue light is having more energy as compared to red light. Am I right? Yes. So first let's understand what is the orientation of electrons. So basically in the ground state, in the S0 state, okay, what happens is that these electrons, they are paired. Okay, they are paired and the spin is anti-parallel. That means that if one electron, that is with respect to this axis, if it is moving in this direction, okay, then another electron will move in this direction. That means one will move clockwise, another will move anti-clockwise. That means anti-parallel electron spin, and the electrons are paired. Now, if light of red wavelength, okay, that means light of lesser energy as compared to blue, if it is incident, the electrons will flow from the ground state to the excited state, and as soon as it reaches to the excited state, that is the S1 state, here what happens is that the electrons are unpaired. Here the electrons are unpaired, okay, and still they are anti-parallel. Still they are anti-parallel. So this state is called as the S1 state, and electrons then return back to the ground state from the S1 state, okay, and this process, it leads to loss of energy. But when you are having a light of higher energy, let's say blue light, when it is incident, it will cause the electron transition to another state, that is not singlet state. So S1 was the singlet state. Now the electron will have transition to more higher energy level states, okay, and this singlet state was short lived, okay. But when you are using a higher energy light, okay, of lesser wavelength but more energy, then you will be leading to the transition of electrons to some triplet states. And in these triplet states, these are like, they remain for more time, because what happens is the transition happens like from singlet, first this, then to triplet, and then to ground state. So there is a longer path that is being covered, okay. So in this triplet state, what happens is that the electron orientation becomes parallel. If both are clockwise or both are anti-clockwise, they will be in same spin, with respect to the axis, and they are in the unpaired state. Okay, because of the high energy, paired electrons gets unpaired. Okay, similar was the case, okay, in the singlet state. So basically in triplet state, the electrons can be there. Okay, so that is how this electron flow is happening to singlet and triplet states as light of different energy is falling. Okay, I hope this concept is clear to everyone. If this is clear, then you can see these definitions. First is your thermal deactivation, that is loss of energy as heat. As electron returns to their ground state, there can be thermal deactivation, that is the loss of energy in the form of heat. There can be fluorescence, that is emission of radiant energy within 10 to the power minus 9, that is nanoseconds, while the electron returns from its first singlet state excited level to the ground state, that is your S1 state. Okay, there can be a phenomena called as phosphorescence, in which emission of radiant energy happens in milliseconds. Okay, that is in milliseconds. Here it was nanoseconds. So in milliseconds, the emission of energy happens when the electron returns from triplet state to ground state. Then it is called as phosphorescence. Fluorescence when they return from singlet to ground. Phosphorescence when electron returns from triplet to ground state. Then energy transfer is transfer of energy between pigment molecules within a photosystem. So basically this part is clear, I guess. So if it is clear, we can solve a question. That is, upon absorption of a photon, a chlorophyll molecule get converted to its excited state when the energy of the photon is more than that of ground state, equal to that of pigment, more than that of ground state but lesser than excited state, or equal to energy gap between ground state and excited energy state. This is the correct answer, because here you require a specific wavelength, specific light energy, red or blue. Okay, that means that the wavelength of the light should be that specific that the molecules are able to absorb it and then reach to the excited state. So basically the wavelength, it should be equal to the energy gap between the ground state and the excited state. Okay, so this energy gap delta E, if light falling with a wavelength lambda such that the energy that is incident is equal to delta E, then only the transition will happen. Okay, and if let's say the energy is more, then it will go triplet and then back to the ground. So that is how it is happening. So the energy should be equal to the energy gap between the ground state and the excited state. Now another question is that after absorbing light, chlorophyll molecules in green plants exist in singlet and triplet states. Following are certain statement on singlet and triplet states of chlorophyll molecules. Singlet state is a short-lived compared to triplet. That is what I told you. Absolutely correct. This is wrong. Singlet state contain electrons with anti-parallel spins, while triplet state carries electrons with parallel spins. I told you that is correct. So correct option is A and C. Okay, this is wrong, opposite statement. Okay, so once you have solved these two questions, you are good to go with the photophosphorylation. Before starting with photophosphorylation, let us understand in detail about the electron carriers. There are questions in exam. The exam is flooded with questions on electron carriers. Basically, these carriers are, we can divide them into two parts. One are for PS2, one for PS1. In PS2, the first electron acceptor, that is pheophytin. Then you have plastoquinone A and B. These are mobile elements. Then cytochrome B6F, which is responsible for proton pump generation. Then you have plastocyanin, that is again a mobile element. Then you have PS1. Okay, this PS1, this has A naught, that is nothing but chlorophyll A. It has A1, that is nothing but phylloquinone. And then A2, that is iron sulfur proteins, which is the FEX, FEA, FEB, which are three iron sulfur cluster protein. It consists of three proteins that I have told you in the lecture in which I taught you PS1, that is the second lecture. Ferrodoxin, you have ferrodoxin. Okay, FNR, ferrodoxin reductase enzyme is there, which helps in the conversion of this NADP plus to NADPH. and NADP, that is your terminal electron acceptor. So the first question, very important question, which many people mark wrong, is that which is the terminal electron acceptor in the Z scheme in the non-cyclic photophosphorylation. The answer is NADP. Okay, and then not FNR. Kindly note, not FNR, it is NADP. Then ATP synthase. Okay, this is responsible for ATP formation. Now I have summarized for you all the electron carriers, their nature, their mobility, their property. This is more than sufficient for any entrance exam you are targeting, CUET, PG, GAT-B, any entrance. Okay, so the P680, also called as PS2, it is basically consists of this chlorophyll A as a reaction center. It is fixed, it is not mobile. It is a strong oxidant involved in water splitting. Pheophytin is a modified chlorophyll A which consists of no magnesium. That is what I told you when I taught you plant pigment, that the chlorophyll A minus magnesium is pheophytin. Okay, so pheophytin is modified chlorophyll A. It is fixed carrier. Okay, and first stable electron acceptor of PS2, very important. Then you have quinone A, that is tightly bound plastoquinone, non-mobile. Okay, single electron acceptor. Okay, the important point to be noted is it is tightly bound to the membrane, and quinone B is loosely bound to the membrane. I told you that it is in thylakoid membrane core, and the thing is that this is a mobile carrier, very important. Accepts 2 electron plus 2 protons, blocked by DCMU, so all things are summarized in this table. Plastoquinone, that is a lipid soluble quinone. Okay, and it is again also a mobile electron carrier, transfers electrons and protons. Plastocyanine. Okay, then we have cytochrome B6F, that is a cytochrome complex, mainly dimeric complex, fixed electron carrier, proton pump, responsible for generation of proton pump, analogous to complex 3 of mitochondria, guys. This is some question that they link with the electron transport chain that happens in our body and the photosynthesis. So that is important. It is analogous to complex 3. Plastocyanin is copper containing protein. Plastocyanin, with this cyanin word only you understand. Okay, it is copper containing. It is again a mobile carrier located in thylakoid lumen. P700 or PS1, guys, this is a chlorophyll A reaction center, fixed, not mobile, strong reductant. Why it is called a strong reductant? Because it has a higher tendency. It is a very good reducing agent. It has a tendency to donate electron to NAD plus and form NADPH. That's why it is a strong reductant. And PS2 is a strong oxidant because it lost its electron and now it is electron deficient, and something which loses electron is a very good, you can say, oxidizing agent. That is something that can oxidize others. Okay, and that they do by donating electron or by giving electron. So it lost its electron to become a very strong oxidizing agent. That's why it is called strong oxidant. Okay, that is the concept. Then A naught. A naught is your chlorophyll A, that is fixed, first electron acceptor of PS1. A1 is phylloquinone, that is vitamin K1. Okay, and that is again fixed quinone electron carrier. Then FES centers, FX, FA, FB. These are iron sulfur proteins, fixed. Okay, these are highly conserved from plants to plants. Then you have the ferredoxin, that is iron sulfur protein, again mobile carrier, very important. Okay, it is present on the stromal side and it functions in cyclic and non-cyclic electron flow. Okay, then you have FNR, that is flavoprotein enzyme, FAT dependent. It is fixed carrier, reduces NADP plus, reduces it to NADPH. Then you have NAD plus, that is a coenzyme, that is mobile, important, terminal electron acceptor, ATP synthase enzyme complex, fixed, operates via chemiosmotic hypothesis. So this is a very important table. You need to learn this table. It summarizes everything for you. Okay. Now, without further delay, let's start with the non-cyclic photophosphorylation, also known as Z scheme, because the electron flow is like a Z. If you look at like that, it is a Z. Okay. So zigzag system also people call it. Basically, here we have the redox potential minus 1.6, 0, 1.6. Okay. So basically what happens is that in this scale, what is happening is, first, like, you are in the light is incident on the photosystem 2, that wavelength of the light will be less than equal to 680 nanometers. Okay, it will be less than equal to 680 nanometers. Then it will excite the electron. Two electrons will pass through phaeophytin, that is the primary electron acceptor. Then to plastoquinone A, plastoquinone B. This is the part that is inhibited by DCMU. Then these two electrons, so once they are passing through the plastoquinone A and plastoquinone B, that is leading to ATP synthesis. How it is leading to ATP synthesis, I will show in detail. Don't worry. First, understand the flow. Then two electron will move to cytochrome B6F complex, which is responsible for proton pump generation. Then these electrons will move to plastocyanin, again a mobile carrier. Then to PS1, then to A0, that is chlorophyll A, then phylloquinone A1, then to A2, okay, and then it will move to the ferredoxin, and there the ferredoxin reductase will cause the reduction of NAD plus to NAD, so it will convert it will reduce the NADP plus to NADP negative negative, that is, it will reduce it, and then it can get some protons from the stroma and can lead to the formation of 2 NADPH. So in this entire process, you are generating ATP, you are generating NADPH, and that is why it is the non-cyclic reaction that is happening, also called as the Z scheme. Okay, so basically in this, what happens is that the carriers which are having a higher redox potential, you can understand from this curve that the redox potential is increasing as from one carrier to another carrier. How it is increasing? That the carrier with a higher redox potential, higher reduction potential, has a more tendency to accept electron. That means more tendency to get reduced. That is what is important to know. Okay, so this is the non-cyclic flow of electrons. Now this flow is in more detail here. So let us see what is happening by zooming into the photosystem 2. So basically, when light is incident with wavelength less than equal to 680 nanometers, it is causing the electrons to it is causing the P680, that is the photosystem 2, to get to the excited state. And once it is moving to the excited state, it returns back to the ground state, it becomes oxidized, and as it is oxidized, it is becoming like a strong oxidant. Okay, and then it is passing the electrons to phaeophytin, that is the primary electron acceptor. Then these two electrons, they are passing to plastoquinone A, then to plastoquinone B, then the plastoquinone B. So basically, since it is a quinone, basically what happens is, if it accepts one electron, then it becomes semiquinone. If it accepts two electrons, then it becomes quinol, that is alcohol from ketone. Okay, so it gets fully reduced. So when this PQB, it accepts two electron, it becomes quinol. So now this is quinol. It accepts some two protons from the stroma, converts into PQH2. This PQH2, it reaches to the cytochrome B6F complex. Okay, and there it gets oxidized. Once it is oxidized, the electrons split the direction into two directions. Electron flow is now moving. One electron will again be accepted by a semiquinone, and it will lead to formation of quinol. So again it will be accepted by semiquinone, okay, and it will form the quinol. So again this quinol will be reduced by accepting protons, again it will be oxidized. So this cycle is called as Q cycle, okay. And then the another direction was, one was the cyclic Q cycle, the electron gets involved in the Q cycle for the regeneration of quinol. The another was that the electron passes through the iron sulfur clusters and then it passes through the plastocyanin, that is a mobile carrier, okay. So now we are passing only one electron, you can see, okay, because one electron got involved in the Q cycle. So if this electron is now passing through the plastocyanin and then it is passing through the PS1, that is P700, okay, and then to the chlorophyll A0, then to the phylloquinone, then to the iron sulfur proteins, okay, FEX, A and B, three proteins. Then it is passing through the ferredoxin, then FNR, ferredoxin reductase, converting the NADP to NADP into 2-NADPH. That is, it is forming first 2-NADP negative negative, and then it is acting as a strong reducing agent. The PS1 acts as a strong reducing agent, okay. And since it is a strong reducing agent, it will take, what will happen is, so basically you can see, since the PS2, okay, wait, yeah, since the PS2 over here, this PS2, it loses electrons, 2 electrons, it will get electron from the D1 protein tyrosine residue, okay, at the 161th position, okay. This will give electron to the P680 and this tyrosine residue will get the electron from the manganese, that the oxygen evolving complex, okay, or the manganese stabilizing protein. So here this will get electron from the MSP and MSP will get electron from photolysis of water. So two water molecules will split into 4 electron, 4 proton in oxygen. Again, proton concentration inside the lumen is increasing because of the cytochrome B6F complex as well as the photolysis of water. And these protons, this will lead to formation of a proton gradient, because you can see in the stromal side, the protons are basically these protons, it moves to the ATP synthase. You have the F0, you have the F1, and because of this movement, this energy proton motive force, it is causing the coupling of ADP to Pi and forming ATP, and this ATP that is being formed in the stroma and the excess protons which are being pumped out, these protons combines with this 2-NADP negative negative and forms 2-NADPH, that is the strong reducing agent. So PS1 is causing the formation of a strong reductant and PS2 is forming a strong oxidant, okay. So this was the overall thing for the Z scheme, how it is working, what is Q cycle, how the electron flow is happening. Now if this part is clear to everyone, there is another representation, similar representation only, pheophytin QA QB, okay. Then this is the cytochrome B6F complex, you have the iron sulfur protein cytochrome F, okay, and electrons moves to plastocyanin, then to this excited state of P700, then to A0, A1, FeSx, FeSa, FeSb, ferredoxin, FNR, then finally reducing to NADPH. That is how it happens. And another thing that you have is a cyclic photophosphorylation. In cyclic photophosphorylation, when the cyclic photophosphorylation happen, guys, this is an important concept that many student misses and they don't understand why it is happening. So it can happen if the light incident is greater than 680 nanometer, then only the PS1 antenna molecules will be active. I have taught you this very clearly in the first lecture itself. If you have doubt, you can see. If you are using the PS2 inhibitors like DCMU, then also cyclic reaction will be happening. To maintain the ATP to NADPH pool, like if you want to increase the ATP to NADPH ratio in the like in the thylakoid basically. So what you will be required to do is to increase the ratio, you want to decrease this and increase ATP production. So for that also, only since in cyclic reaction only ATP is formed, that is why cyclic reaction happens, okay, to maintain this ratio. Okay, that is the primary reason. So basically here this is a pictorial representation that when light is incident, this is the reaction center, antenna molecules, they transmit the light. Finally, it moves to reaction center, then to primary acceptor, then to ferredoxin, and then from ferredoxin to plastoquinone, cytochrome B6F, ATP synthesized, then to plastocyanin and back. So let me tell you in detail. This is the P700 PS1. So first electron will move to the A naught, correct? That is the chlorophyll A. Then it will move to the phylloquinone. Okay, then to the A2, that is the iron sulfur protein cluster. Consists of three proteins, FeSX, A and B. Then to ferredoxin, and from ferredoxin it will not move to the FNR, okay, or the NAD plus. It will now go to back to the plastoquinone A, B, and ATP will be synthesized, then to cytochrome B6F complex, and then to plastocyanin, and again back to PS1. So this is the cyclic flow. Now if you want to understand it in detail, let's see. So since this is PS1, okay, electrons are moving from A0 to A1 to A2, then to ferredoxin, and then not to NAD plus, but it is moving to the that is the quinone B, that is the plastoquinone B, okay, and electrons are causing it to form the quinol, okay. So to semiquinol, the electron gets added and it forms quinol. Then from stroma, two protons it reacts with and forms PQBH2. This PQBH2 again it leads to oxidation. Then it converts into quinone B, again plastoquinone B, and electron again deviates into the Q cycle, okay, and the another electron it passes through the cytochrome B6F complex, then to the PC, that is plastocyanin, and back to the P700. So this is the cyclic photophosphorylation or the cyclic flow of electrons. Now let's look at the energetics. Let's look at the calculations. So for the photolysis of water, 4 electrons, 4 protons, oxygen is being produced in the PS2, right? When PS2 was active. But so this is in case of cyclic reaction, what is happening is you are getting 8 protons inside the lumen, because in cyclic reaction, see, you are not getting photolysis of water. Since you are not getting photolysis of water, these protons gets cancelled. They are not there. Okay. Only thing you have to consider is that this electron is moving and one electron is leading to pumping 2 protons into the lumen. So if I have to compare it with non-cyclic photophosphorylation, let's say if I take 4 electron, if I multiply this equation by 4, then 4 electron will pump 8 protons into the lumen. And if 8 protons are being pumped into the lumen, then what will happen is that for every 4 protons you can form 1 ATP. Okay, this is the ratio. So if you are having 8 protons, then you can form 2 ATP, correct? So in cyclic photophosphorylation, you can form 2 ATP, no NADPH, and PS2 acts as a strong oxidant, PS1 acts as a strong reductant. This I told you. Then if we extend this further, in non-cyclic what happens is, in non-cyclic you have the 8 protons from the flow of the cytochrome B6F, the proton pump, and then you also have the 4 protons from photolysis. So total you get 12 protons. Out of those 12 protons, for like you can form 3 ATP and 2 NADPH are formed in non-cyclic. In cyclic you get 2 ATPs. And in terms of photons, if you want to look at this reaction, Then for splitting two water molecules, you require light energy equivalent to 8 photons, E equal to NH nu. Okay, so basically you require 8 photons. You require 8 photons, okay, for hydrolysis of two water molecules. Okay, so this is in terms of photons. Now there is a very important experiment that was being done, and questions can be asked based on this experiment, that is acid base phosphorylation experiment carried out by Jagendorf and co-workers. In this experiment, you have a buffered medium with a pH 4. So one concept is that if pH is less, proton concentration is high. So if pH is less, proton concentration is high. You added the chloroplast thylakoids, guys. You added the chloroplast thylakoids. Then you equilibrate it with the buffer. So what will happen? The lumen will be enriched with protons. Lumen will be enriched with the protons, and since in this you don't add any ADP or PI because this is in vitro, there will be no ATP synthesis. Now, when I put this into a buffer media of pH 8, that is pH is more now, and H plus ion concentration outside is lesser. Okay, and this all is happening in the light condition, guys. Now, the question is, can ATP synthesis happen at dark? The answer is yes, it can happen in dark. Okay, it can happen in dark. So the thing is that in dark, I provided the ADP and PI, and now this can be combined because now the inside lumen concentration of protons is high, outside H plus concentration is less. The protons will move from lumen to stroma outside, and it will cause the formation of ADP plus PI, and it will form ATP. So basically, this was the experiment done in vitro to verify this fact. Now this is the electron flow, basically how the electron flows from P700 basically to chlorophyll A, phylloquinone, then this, and in PS2 they flow to pheophytin, plastoquinone, QA, QB. So this was the electron flow, and this is the table for the difference between cyclic and non-cyclic photophosphorylation. So photosystem involved is PS1, P700. Here in non-cyclic you have both photosystem 1 and 2, right? Because here you saw that Z scheme, right? So everything was involved, whether it is 680 or P700, both were involved. But in cyclic one, only this was involved. So water is not required for cyclic, water is required for non-cyclic. Photolysis of water is absent here, here it is present. Electron flow, electron travels in cyclic manner, electron travels in non-cyclic linear Z zigzag manner. ATP synthesis, ATP produced in both. NADPH is not formed in cyclic, but it is formed in cyclic. How many NADPH are formed? 2. How many ATPs are formed in the non-cyclic? 3. How many ATPs are formed in the cyclic? 2. Okay, if you are able to answer this much fast, you are good to go. Oxygen evolution. Oxygen is not involved because there is no water splitting. Oxygen is evolved. Effect of inhibitors, affected by DCMU, and non-cyclic, it is affected by paraquat. Okay, occurrence predominant in bacteria, the cyclic one, and the non-cyclic is predominant in all green plants. This is you have to remember. Okay, so this was the differences. Now I guess we are good to go with some questions. So which protein component is mobile in between PS2 and PS1? So this is not a mobile component, but these are mobile. But the question is that which protein component is mobile? Okay, now if you are not able to recall, you can look at the diagram that I have shown. Okay, so here you can see this is PS1, this is PS2. Okay, in between PS1 and PS2, the carrier, the mobile carrier, okay, protein carrier that is involved is the plastocyanin. Okay, these plastoquinone also In plastoquinone you have quinone A, quinone B, out of which quinone B is mobile only. Okay, so the thing is that if you see the options in the question, okay, the ferredoxin that is present in the stromal side, that is again mobile, but that is not between PS1 and PS2. So this is not the option, and this plastocyanin is the correct option. This is also not the option because QB is mobile but QA is not. Okay, so this is what you should know. Then the role of carrier protein light reaction is to transfer energetic electrons from PS2 to PS1. Pumping of protons, no. Formation of ATP, no. Transferring high energy electrons from PS1 to PS2, no. Answer is 1. Okay. Now the donor and acceptor of electron for cytochrome B6F. In cyclic, you remember that the cytochrome B6F which we have, so the acceptor was PC and the donor was plastoquinone, which was donating the electron, splitting into two directions. Okay, so this was the thing. So here the answer is PQ and PC. Okay, then chemiosmotic hypothesis. Basically, you should have an analogy between the chloroplast and mitochondria. So in case of chloroplast, in the lumen you have high proton concentration. In the stroma you have lesser proton concentration. Reason for high proton concentration, repeat with me, photolysis of water. Then you have that oxidation of the PQH2 to PQ. That is another reason. Okay. And so because of this there is a formation of a proton gradient, okay, from lumen to stroma, and because of this only the ATP is getting synthesized. This proton is moving over here, okay, and ATP synthesis is able to synthesize the ATP, right? So that is what is happening in chemiosmotic hypothesis. Or in chemiosmosis, basically what happens is you have the electron transport chain, you have different complexes. So when the electron flow through these complexes, in the intermembrane space you have a higher proton concentration. Okay, so that is what is happening in the mitochondria. Okay, so there it is due to the movement between these complexes. Here it is with the stroma and the lumen. That was a simple analogy you should remember. You should always study by connecting things, that way you learn better. Okay, and similar thing is being represented over here. When these protons move through the ATP synthase, you get the formation of ATP. So high electrochemical potential gradient to low gradient of protons, high proton, low proton. Okay, let's solve another question, guys. Plant dissipate excess light. So these questions are from the previous lectures also. Plant dissipate excess light energy as heat so as to protect from photooxidative damage. Mechanism is known as, the mechanism is known as non-photochemical quenching. Okay, I told you that there is something that is called as a thermal dissipation. That is the first line of defense. Line of defense. Line of defense. Okay, first LOD. That was the non-photochemical quenching. Okay, photo inhibition and all this, they comes in the second line of defense. Okay. Okay, so then another question. The photo intensity captured by green plant is used in the following processes: photosynthesis, generation of heat, production of toxic products like superoxide, damage to D1 protein of PS2. Based on the above fact, photo inhibition will happen when the entire proton intensity, no. Excess photon is used for heat generation, no. Okay, photo inhibition is what? It was the damaging to D1 protein. It was the damage to D1 protein of PS2. Okay, so here excess photon intensity leads to damage of D1 protein. This is the correct option. If we look it at further, which of the following mechanism is not involved in providing photoprotection to plants? So I told you that in photoprotection you have first line of defense, you have second line of defense. In first line of defense you can have thermal dissipation, you can have xanthophyll cycle. Okay, and then you can have another thing that can be in the second line of defense. You can have something called photo inhibition, in which you have a D1 protein degradation. But the photolysis of water, it is not involved in providing any protection. So that is the answer. Okay. In chloroplast, site of coupled oxidation reduction reactions is the site of coupled oxidation reaction. Since all these proteins, they are in embedded in the membrane, then it is thylakoid membrane. That is the answer. Okay. Then there is a electron flow. Electron transfers through carriers organized in the Z scheme following are indicated. So from P680, what you have the pheophytin. Then you have this is the primary electron accepted. Then you have plastoquinone A, B, cytochrome B6F, then plastocyanin and P700, correct. P700 you have first A0. You can learn from the number also, 0 and 1. So B and C. Then FeSX, A and B, and ferredoxin. After that you have the NADP plus, that is the terminal electron acceptor, remember it. Okay. Then in an experiment, spinach chloroplasts are illuminated in the absence of ADP and PI. Then the light is turned off and ADP and PI are added. Identify the correct statement regarding the status of ATP synthesis. So what will happen? Basically, in spinach chloroplast are illuminated and the light is turned off and then it is added. Okay, ATP and PI. Now, the rate of hydrolysis of ATP will be enhanced in dark. No, because with time, as time increases, what will happen? This concentration gradient that is formed, because in the buffer of pH 8, the H plus ion concentration was less, but it was somewhat H plus ions which are present. So since with time, what will happen? This concentration gradient will decrease. And as concentration gradient will decrease, the movement of protons from the lumen to the stroma, it will decrease, and it will lead to a decrease in ATP hydrolysis, not increase. Okay. So first thing is there will be decrease in not ATP hydrolysis, decrease in ATP formation also. Okay. So the option is anyways wrong. Okay. No synthesis of ATP at all in the dark. No. ATP cannot be synthesized from ADP and PI without the addition of appropriate enzyme, but enzyme, like, no need of any enzyme. No, you have ATP synthase. So wrong. ATP synthesized for a short time in the dark till you have the proper concentration gradient. So answer is 4. Okay. Last question for today, and then we are done with this. So A is an inhibitor of chloroplast function. Production of oxygen and synthesis of ATP are measured in illuminated chloroplast before and after addition of A. What is correct for A? What is the mechanism of action of A? So this I am drawing a line. This is before, this is after A addition. So before and after, oxygen production remains the same. If oxygen production remains the same, that means electron flow is proper. And if electron flow is proper, that means NAD plus reduction is proper. That means your Z scheme is working fine. The Z scheme or non-cyclic photophosphorylation, that is working fine. Here you can see that before A addition, ATP synthesis was high. As soon as A is added, that declines. So if it is declining, that means there is a inhibition of proton gradient. There is something to do with the proton gradient. Okay. Something to do with the proton gradient. So it is getting inhibited. That's why ATP synthesis is decreasing. So A inhibits proton gradient and the reduction. No, inhibits proton gradient but not the reduction of NAD+. That is the correct answer. Okay. So that is how you can think of it. So proton gradient mainly for formation cytochrome B6F is responsible. So this graph will correspond to cytochrome B6F, and this graph can be corresponding to the PS2. Okay. So this was the question, final question. So we have covered cyclic and non-cyclic photophosphorylation in detail. All the electron carriers we have studied, the differences we have studied, and everything like in detail we have covered. So I hope you can answer all the CUT GAT B questions with this lecture. So thanks a lot for joining everyone.